Good morning. Thank you so much for joining us for our first Bio-Coronavirus Collaboration Summit. We're very excited that many of you could join us today. I'm Phyllis Arthur, the Vice President for Infectious Diseases and Diagnostics Policy at Bio. I wanted to actually just welcome all of you. We have a really interesting and important summit going on over the next 2 days with a lot of great speakers this morning. First, I'd like to actually ask our Bio CEO, Jim Greenwood, to make opening remarks. Jim, please.
Well, thank you, Phyllis, and good morning, everyone, and thank you so much for joining us. Hope you're all safe. From the beginning of this crisis, it was clear to the Bio staff and to our board and to our chairman, Jeremy Levin, that Bio is the organization in the world that would be best constituted and positioned to organize and to connect the biopharmaceutical industry so that we can accelerate the research and development and coordinate that development as this crisis. Do we want to minimize redundancy, maximize cooperation and collaboration? One of the first things we did is I contacted our entire 700 members, asking which of them were already involved in either diagnostics or therapeutics or vaccines or wanted to become more involved and collaborative. 45 of our member companies responded affirmatively. We've reached out, we've produced pre-reads for them, we've connected them to government organizations and authorities as need be. And then, of course, we've organized this summit. I'm delighted that so many of you could participate. We are very, very honored to have Dr. Birx, Ambassador Birx, and Casper and the ASPR, Dr. Kadlec with us, as well as I think Scott Gottlieb and Mark McClellan will be joining us. I want to also thank George Scangos, Dr. Scangos of VIR, formerly of Biogen, formerly of Select there. It's his lifetime career in technology. We offered to chair this effort. He and his staff have been instrumental in working with my staff and all of the companies as we go forward with what we know only hope has for this provide solutions for this deadly pandemic. So with that, I'd like to thank you again and turn it over to Dr. Scangos. George.
I'm CEO of VIR, a biotechnology company actually focused on infectious diseases. My colleagues at Bio have asked me to take on the leadership coordination of the industry's efforts to fight this new coronavirus outbreak. So look, it is a time of national, global uncertainty, huge anxiety. Everybody's concerned about whether or not they might actually be infected with COVID, what the outcome will be. The future in general seems very uncertain now as opposed to what it seemed like a few months ago. And so it is an incredibly stressful time. I have been impressed, actually, with the response that people have developed towards this epidemic. And I think that starts with, doctors and nurses, and many of us have family members and friends who are on the front lines dealing with patients, putting their own health at risk to do what they need to do to keep other people alive and get them on the road to recovery. So those people are remarkable, and I think we owe them an incredible debt of gratitude. Within the biotech industry, I've been known for a long time that the industry is full of people with the best intentions who have devoted their lives to bringing forward medicines that can improve the lives of other people, that can save lives in many cases. And I think this this crisis now has brought that out to the forefront. So many companies have stepped up. And these are biotechnology companies. These are not companies largely with huge profits. Many of them are pre-commercial with very tight budgets. They are offering to do what they can to contribute their technologies, their compounds, whatever it is they have, to try and work together to find some solution for this pandemic. So I think this has brought out and highlighted the very best of our industry. There have been 45 member companies who have stepped up, who have indicated they have some technology, some compounds that might be useful for the for treatment of the COVID-2. I have a lot of different approaches, a lot of different technologies, a lot of different needs. Right? A lot of the companies are small and don't have the complete set of technologies that they need. So part of the goal of this summit is to bring those people together. It's a 2-day summit, and there are 3 really overarching goals. First, we want to inform everybody about what is known about the virus, what's known about the outbreak, what projects are underway, what knowledge gaps there are, what policy challenges there might be to address effective countermeasures. Right? Second, we want to tackle the best approaches, identify them, tackle them. Could include therapeutics, vaccines, and other ways to prevent the disease, diagnostics, other tools to respond. And third, we want to talk about collaboration strategies. How do we get companies together? How do we take companies that would complement each other and encourage them and facilitate their interaction with each other so they can work together to bring forward important new therapies or preventatives. So for all of you bio companies, no matter what stage of R&D you're in, or whether you're here from industry, academia, government, hopefully this is the right place, and hopefully this will be a productive use of a few hours. I think everybody's busy, try and make this as valuable as we can. So look, to that end, I wanted to note the launch of BIO's Coronavirus Hub. This is an online portal where companies that have critical supplies and resources or have critical needs can share them and hopefully find a match with others who are complementary. We've seen requests from medical research centers and testing developers for supplies, such as RNA kits, nucleic acid isolation kits, N95 masks, respirators, etc. We already have sent out notices, and I think those have gone out nationally and also locally, certainly here in San Francisco and other places. So encourage everybody to help protect our population and certainly our healthcare providers. So the hub is a place where you can make these kind of requests, donations, and capabilities. So we get a full agenda. We truly hope this is just the beginning of this effort to build greater collaboration and coordination across the industry. So at the end of the summit, we want to have a list of action items to pursue during the coming days and weeks. So whether it's speeding new therapeutics or preventatives, addressing some of the R&D or manufacturing and scale-up challenges that we may face, or any number of other topics that may come up over the course of the 2 days. So we've got a lot of ground to cover, many questions that need to be answered. And so I'm looking forward to a robust meeting. And so again, thank you all for joining and this virtual summit and for being part of the important efforts to try and do something to head off this pandemic. So with that, I'd like to introduce a few distinguished guests for remarks. First, we'll be hearing from Ambassador Deborah Birx, who's been at the forefront of the national response here in the US. I've seen Dr. Birx on CNN several times over the past few days, and really want to thank her for all of her efforts. And she has the coronavirus response coordinator in the White House. Second, Dr. Robert Kadlec from the Department of Health and Human Services, where he's Assistant Secretary for Preparedness and Response, will address the summit after Dr. Birx concludes. So with that, let's move on, and let me introduce Ambassador Birx.
Thank you, and thank you for getting everyone together virtually and being responsible and practicing social distancing. I really appreciate it. I come out of the world, we were very dependent on advances in biotechnology, and I just want to thank all of you for the work that you have done and continue to do against other infectious diseases, whether it be malaria, TB, HIV around the globe, and this current pandemic. I really want to emphasize that what we are looking for right now is this virus, as opposed to many other viruses, is quite stable. And what do I mean by that? Our key antigen sites that we would want to utilize for a vaccine or for monoclonal antibody work is more stable than any of our other flu-like viruses or other issues. So I think the targets have a level of stability, although we're tracking very clearly whether there's any molecular migration, because obviously it's an RNA virus that does have the potential to transform. So we're very interested in what can be developed that can be used as a pre-exposure prophylaxis for frontline healthcare workers, a post-exposure prophylaxis that can be used from individuals in households that have a family member that was diagnosed, and of course, a whole array of therapeutics. But if this virus persists, if our bell-shaped curve looks like flu or SARS, we know that there will be a grace period potentially in the summer. It also means that it could come back in the fall as opposed to SARS. And I think having a full repertoire of pre-exposure prophylaxis, post-exposure prophylaxis, full line of therapeutics going into the next fall, if it happens again, the fall of 2021, where you would have full vaccine capacity. And so I think based on the work that you are going to do and your ability to collaborate with one another, positions us not only for the moment, but for the next 6 months, the next year, and the next 18 months. And we're really thrilled about the work that you're doing. Finally, just to return to something that's very important to me, having worked on, I came out of the therapeutics and vaccine work of HIV before I ended up doing implementation through PEPFAR and global health. But diagnostics obviously still is going to play an absolutely key role in this. Having a point of care, true point of care rapid test would be game changing for us right now, whether it's antigen detection, early antigen detection, or early IgM, IgG detection. These are issues that would really help us tremendously. I want to thank the commercial laboratories who rapidly adapted all of their HIV viral load technology to this question, to this work. And the tests that you're seeing now, the 90% or 80% of the tests that we have done over the last 7 days has been because of the biotechnology world and the commercial innovative world, private sector helping us to get there. So I will be relentless in my requests from you and to you. Point of care diagnostics, both antigen detection and antibody detection, absolutely key. Pre-exposure and post-exposure prophylaxis, therapeutic options, and then penultimately the vaccine, all working on a very tight timeline. So we have improved diagnostics and potential therapeutics based on off-the-shelf or new innovations, plasma, or even hyperimmunoglobulin could potentially be rushed to market within the next weeks or so if we all work together. And then broader options around monoclonal antibodies, small molecule research, and the ability, I think our target in this case, and because of its linkage and its complementary to SARS, have really helped us in a rapid development phase. So I thank all of you for the work that you're doing. My expectations are high because you've delivered for us before, and I really know that you can deliver for us again if we have this consortium that works together for the benefit of the American people. So thank you for what you're doing. Thank you for the organizers who pulled this together. I think it's really extraordinary, and I appreciate your innovation and how to bring people together in new ways. Thank you.
Okay, thank you very much, Ambassador Birx, and thank you for taking time out of your schedule to be with us today. I know you're incredibly busy these days, and we certainly appreciate your being here. Let's turn to Dr. Robert Kadlec from the Department of Health and Human Services, where he's Assistant Secretary for Preparedness and Response.
Thanks, George. And to Jim and Phyllis, greetings, and to the membership here, thank you very much. Clearly, in the evolution of this event, when we last talked, it was more of a theoretical set of circumstances we were discussing, and obviously that's changed dramatically. I'll say two things. We're confronted with the worst best case we could have, I think, insofar as something that's highly transmissible, but yet at least for a segment of the population, a pretty significant virulence. But for the majority of the population, something that is mild. But the dilemma is it's still a challenge to our healthcare system and ensure that that doesn't get crushed by the sheer number of people who may seek care and need particularly intensive care as we go forward. I just want to give you a little sense of what ASPR is doing. Clearly, we are bolted to the side of FEMA at the present time, and basically providing them the kind of like strategic public health and medical guidance, so they can execute through the national incident management structure how to best affect a national disaster that's unfolding, at least regionally right now, but likely to basically roll across certain parts of the country in the coming weeks and months. I do believe that at the present time that we're kind of hanging in there, but I have to tell you, first of all and foremost, that we're really operating against, I think, a very sound strategic additional strategy called the 4 S's that we're trying to do that would hopefully do 2 things: save lives and infrastructure of the country. I'll give you a quick overview of that and then maybe talk about some of the specifics. But critically, we're trying to shield the vulnerable. And again, when we consider the vulnerable, we just need to look at the papers and understand, as skilled nursing facilities, as those in Washington State and other parts of the country, have been devastated by this virus. So saying, how can we basically ensure that the people who are at most risk are protected as best we can from the effects of this virus or exposure to this virus? That is clearly a work in progress. However, it has manifests itself in a number of ways. We try to work with the associations and directly with skilled nursing facilities as well as elder care, and the work that the dialysis companies are doing is pretty extraordinary to keep their dialysis clinics open and virus-free. Second S is really sheltering. This is everything that CDC is doing, mitigating the effects on the general population and increasing the transmission, which again is another strategy to protect the vulnerable, but also ensure that the numbers that occur in any community do not overwhelm the healthcare system. The third one is saving the sick, which is really about 2 things. One is hopefully segregating the people with the coronavirus away from the regular part of the healthcare system, so we continue to serve people who are having babies, who are having heart attacks, other medical emergencies who need care at the present time, and as well as kind of avoiding, if you will, that surge, particularly in intensive care, that would be crushing, as we've seen in Italy. And then the last one is most important, all about logistics and sustainable supplies. This applies to not only personal protective healthcare workers, but also for the medicines that you and the very, very things that you make every day that are vital to keep America healthy and alive. So we're focusing on all those 4 lines of effort. We're trying to do this as a means across not only just across the federal interagency, but whole of government, whole of nation, to basically do this. Now, I think George mentioned in the beginning about the uncertainties here, but I'll tell you some certainties, which is what I've seen firsthand, which is the certainty of individuals and groups like yourselves that have basically self-organized to come together to try to find ways to basically help the country in this need. And that is something extraordinary, and I just applaud membership in general and leaders basically organized quickly in this realm. Obviously, things don't happen in this field in science and in pharmaceutical development fast, but I think from Bar to hear shortly from Rick Bright that there is a lot that we're doing with you and other companies in the outreach we've had. Probably over 1,800 companies, dated that number, who have basically been involved in trying to basically find ways to basically contribute their science, their potential product line, their platforms to basically address talent. And I think that is an extraordinary testimony to not only why America's great, but America's good. And I am very certain of this, which is that we will survive this event. We will be sadder, maybe wiser to this. But the very things that we as an organization, you as an organization, and we as a collective entities have been talking about over these decades since basically 9/11 is basically, again, this worst best case where we can basically not only hopefully stem this thing, and I'm convinced that we'll come up with not only great diagnostics that have been defined already, and increasingly more that are giving us point of care capabilities, but potential therapeutics that will in the short term. Obviously, we need to work tirelessly on this. We can't be faint or fatigued, but we have to be determined in this effort. So with that, I just want to say thank you very much for this opportunity to participate. And I really think that at this point in time, the best of what we have to offer is yet to come, as collectively as a nation, and certainly as you as a group of companies. And I think you'll hear from Rick about a lot of the things that he and his counterpart, the FDA, also non-government organizations are doing in this cause, in this fight. So thank you.
Mm-hmm. Okay, well, thank you, Dr. Kadlec. I think for those encouraging remarks. And I appreciate, again, your being here and taking time out of your busy schedule to join us as well. So now I would like to turn to our next speakers. First, we'll be hearing from Dr. Richard Whitley, who's a distinguished professor of pediatrics and vice chair of the Department of Pediatrics, co-division director of pediatric infectious diseases at the University of Alabama at Birmingham. And then second, we'll be hearing from Dr. Susan Gerber, who's chief of the Respiratory Viruses Branch, the Division of Viral Diseases, National Center for Immunization and Respiratory Diseases at the Centers for Disease Control and Prevention. Just a reminder that there's an area on the screen where you are viewing that on the session that you can submit questions. So after these remarks, we'll have a Q&A session. So with that, let me turn it over to Dr. Whitley.
Just as background, I'm going to take a 35,000-foot view of what we know about the pathogenesis of coronavirus infections, and I'll leave the epidemiology to Dr. Gerber. I think it's important just to remember that coronavirus infections have been with us forever. Coronavirus infections in general are the second most common cause of upper respiratory tract infection, namely the common cold. But it's been in the past 20 years that we've seen both the evolution of SARS and MERS, and now SARS-CoV-2, with increased pathogenicity. But just remembering that these viruses are with us all the time, and that we're now seeing more significant disease caused by coronaviruses, I think is important. So the lessons that we learn from coronavirus replication provide insights not only in the development of antiviral agents, but also in the evolution of diagnostic and vaccines. We just think about SARS-CoV. Some of the characteristics of the disease are this involvement of the lung with a deep cough that is persistent over time, and that has been one of the noticeable symptoms of the disease that patients report. So why is this cough important? Well, we need to just remind ourselves that SARS-CoV-2 uses what we call as an ACE2 receptor in the lung to allow the infection of lung cells. ACE2 is an exopeptidase that allows for the conversion of angiotensin 1 to angiotensin 2. It's also present in both heart and kidney cells as well, and may be a targeted site for the development of new antiviral agents, if not for the development of vaccines. As we think about what happens with human disease, SARS-CoV-2 is different than some of the other circulating concomitant viruses that we see in the community at this time. So the two that we worry about the most are influenza, which has had a protracted season this year, and rhinovirus infections, on top of which we're seeing seasonal allergies, which can mimic some of the infections that are common amongst all of us. Characteristic of SARS-CoV is this deep cough, a slowly evolving fever, namely a gradual onset with an incubation period between 2 and 14 days, averaging 5 days, and I'll let Susan get into that in more detail. Infection is associated with fever and characteristically shortness of breath, and it's the shortness of breath that distinguishes it from influenza, because with influenza we can certainly see fever, which is common, we'll see a dry cough, we'll see rapid onset as opposed to an insidious onset and a sore throat. Both diseases are associated with fatigue, and aches and pains being more common with SARS-CoV-2. In contrast, when we think about just colds, we know they're associated with runny noses, sneezing, rarely with fever, sometimes with cough, but they're a much milder illness. And obviously, allergies are seasonal in occurrence and associated with runny nose, sneezing, swollen red eyes, and significant discomfort. It goes back to the point that Dr. Birx made, and that is, it really is important to have diagnostics that are at point of care using what we know about the replication of the virus, whether it be the spike protein or whether we use receptors as that method of diagnosis, so that we can distinguish between these entities. Probably more importantly is the epidemiology, and I've avoided talking about asymptomatic shedding because I'm sure Susan's going to bring that up, and probably it's wise if I just turn it over to her at this juncture.
I'd like to thank everybody for your patience while trying to get through this, and I'd like to thank the organizers for inviting CDC and Dr. Whitley for his comments. I'd like to also say that CDC is aggressively responding to the outbreak of COVID, which is unprecedented. And what I first spoke about was the emergence of a virus identified in December of 2019. And it was originally reported to be associated with live animal markets, but subsequently discovered more cases were reported that clearly did not associate with the animal market. This indicated spread. I'd like to talk about where we are with numbers, and these are as reported as of March 23rd. Globally, there have been reported to WHO 292,142 cases with 12,786 deaths. These are among 186 WHO member countries. Domestically, CDC is reporting as of March 23rd, yesterday, 33,404 confirmed and presumptive U.S. cases. And this is among 55 jurisdictions, which include all 50 states and New York City, Washington, D.C., Puerto Rico, and the U.S. Virgin Islands. 400 deaths have been reported in the United States. Now, CDC is also adapting and leveraging some of the agency's existing surveillance system to better track COVID-19. And this will allow us to understand circulation of the virus in different parts of the country and be able to understand better the epidemiology. We know that this virus is capable of sustained community transmission. We do know that it appears to be spread through close contact and secretions, droplets, and possibly there may be a component of touching secretions and putting then inoculating the face. In terms of some of the ways it's transmitting, we still have a lot to learn about the new virus. To address Dr. Kennedy's comment about pre-symptomatic transmission, there is limited information that indicates that it may be possible that there is a TB transmission or spread before a person recognizes their symptoms, but a lot more work needs to be done to completely understand how this virus is being transmitted through people with different symptoms or lack of recognition of symptoms. Clinically, there's a wide range of illness that has been reported associated with SARS-CoV-2 infection, and these illnesses range from mild, moderate to severe infections. Mild infections may include cough, fever, and shortness of breath as the disease might progress. And the disease may result in acute respiratory distress syndrome, multi-system organ failure, and death. So there is a wide range of symptoms reported, but in particular, older people and people with severe chronic conditions should take special precautions because they are at higher risk of developing serious illness from SARS-CoV-2 infection. In addition, the incubation period has been appears to be anywhere from a range of 2 days to 14 days. The median incubation period from some published data would indicate somewhere around, at least in many studies, some studies, around 5 days. But again, more information is being collected to clearly understand the incubation period range and median. I would also like to say about this outbreak that there's not a clear answer for how long this outbreak lasts in other countries or in the United States. And the duration of the outbreak is dependent on many factors, which don't yet have enough information to address. We are still understanding the effect of social distancing and travel restrictions, the extent to which sustained transmission occurs in other countries, and still we're learning about the natural history of the virus and understanding is there seasonality associated with this virus like other common human causes that Dr. Whitley mentioned, or not. So we have a lot of questions that we pursue immediately. Also, we need to understand the duration of immunity after acute infection and more information about transmissibility and severity of clinical infection. Now, projections will be possible as information becomes available, and CDC does have a modeling team that is collaborating with external groups to ensure timely forecasts. And just a few words about there are there are 4 common human coronaviruses that circulate: 229E, OC43, NL63, and HKU1. Primarily, those infections are usually recognized clinically in children, and they primarily circulate in the wintertime, but circulate all year round, but less so in the summertime, at least in the United States in a temperate climate. There have been reported SARS, which was first identified in 2002, last known case of SARS in 2004, and MERS-CoV, which was actually first identified associated with the Canadian Peninsula in 2012, which there are still MERS-CoV cases to this day. Those infections associated with MERS are also range from mild to severe illness, but more severe illnesses have been reported. But largely all cases remain associated epidemiologically with the Arabian Peninsula. So back to SARS-CoV-2 appears to, unlike SARS-1 and MERS-CoV, does actually sustained community transmission. So a few words about social distancing. Everyone can do a part to help respond to the On March 16th, President Trump at the White House Coronavirus Task Force issued new guidelines to help protect Americans during the pandemic. This initiative is called 15 Days to Slow the Spread, and it lays out guidelines for a nationwide effort to slow the spread of COVID-19. It also calls for the implementation of measures to keep social distancing between people at all levels of society. And this is a massive proactive preventive response to COVID-19, and it aims to slow the spread and blunt the impact of this disease on the United States. And all segments of the U.S. society have a role to play at this time. People across the country are asked to stay home as much as possible and otherwise practice social distancing. And this includes canceling or postponing gatherings of more than 10 people and closing schools in some areas as determined by local and state governments. People have asked about guidelines for people outside, and the guidance remains the same, which is about 6 feet apart, coughing and sneezing etiquette, and that is important. There is no vaccine currently available as this COVID-19 and now approved antiviral treated at this time. However, there are several FDA treatments that help ease the symptoms of the scary environment. So despite the fact that there's no vaccine or treatment available that's approved at this time, there is mounting evidence through journal articles that quite effective measures, both when used alone and in combination measures. And these recommendations clearly work best in context. And while the new initiative and guidelines are recommended for the next 15 days, and we're about halfway through, government leaders will continually reassess as of the outbreak in the United States, and recommendations change as time goes on. I would like to also just a few notes about CDC Travel Guidance. CDC is constantly reviewing and updating travel notices recently, and as of March 23rd, CDC has issued 9 additional Level 3 travel notices for Australia, Brazil, Canada, Chile, Israel, and Pakistan, Thailand, and Turkey. And on March 19th, the U.S. State Department posted a Level 4 global health advisory warning travelers do not travel. And on March 19th, CDC posted after travel precautions for travelers from countries with widespread, sustained, or ongoing transmission who arrive in the United States. Now, CDC does not generally issue advisories for domestic travel. However, the CDC website does include a consideration for traveling in the United States. And I just refer everybody back to the website, and thank you for your attention.
Thank you, Dr. Gerber and Dr. Whitley, very much. I wanted to let everyone know, remind you that underneath your virtual webcast where you see the pictures, there's actually a place to type a quick question, and someone will be feeding them to me to share with either Dr. Whitley or Dr. Gerber. Dr. Gerber, there was actually a discussion going on online about the cases in Washington State and the fact that some people were testing negative, but they actually were also talking about a symptom they were seeing of redness above the eyelid as potentially an indicator of having COVID-19. Have you heard of this as a symptom that people should look for? Dr. Whitley, you may be able to respond as well.
Sure, thank you for the question. I would also like to remind everybody that although there is circulation of SARS-CoV-2 in the United States, there are also other respiratory viruses that are also in the United States at this time, and some of the circulation of other respiratory viruses can be accessed also on the CDC website through the National Respiratory Virus Surveillance System. So, talking about somebody who tests negative for SARS-CoV-2 infection, it is unclear if this symptom, redness around the eyelid, could be attributed to SARS-CoV-2 or potentially another pathogen that could be a virus or bacteria. And so we're still collecting the symptom information from cases, both mild and severe, to better understand the range of symptomatology associated with this particular coronavirus. So that, we're still working on, but, in terms of just the big picture, there are still respiratory viruses circulating, other ones, in addition to SARS-CoV-2.
Thank you.
Thank you.
We had another question. I'm sorry, I'm looking off screen to look at the question, make sure I get it right. So I think everyone's interested in understanding how much we should be testing mild or moderate cases in order to get a better idea of the denominator of how many people are infected. What do you both think about the need to test mild and moderate or even asymptomatic cases in order to better understand how many people are infected and the actual mortality and morbidity and mortality? Maybe Dr. Whitley, you have some comments on that, and then we'll go to Dr. Gerber.
I mean, the major limitation to testing a lot of people is the lack of availability of diagnostic tests. In the state of Alabama, we've had a very difficult time getting people tested, even though many of them have wanted to be tested. And as a consequence, testing is with physician guidance and with physician orders. Even today, March 23rd, we still have an inadequate number of tests to evaluate people who are even relatively symptomatic. Once that becomes more readily available, it would be nice to test more people who have milder symptoms. And I don't necessarily mean it from the standpoint of the natural history of disease, which is important, but also from the standpoint if we develop therapeutic interventions, whether it be antibodies or drugs or whatever, we may be able to give them to people in the early stages of disease before it becomes more advanced. But right now, in my state, we don't have available diagnostics to help us work through that problem. So we're limited to symptomatic people.
Dr. Gerber, your comments?
Right now, we believe healthcare providers can order testing for patients of having COVID-19 for using their judgment. And CDC's current guidance for the evaluation of laboratory testing does provide for this clinical discretion in determining whether a patient should be tested. Clinicians around the country should consider the local epidemiology of this community transmission and also the clinical guidance on identify key patients for the prioritization of testing, which do include hospitalized patients, healthcare workers, and patients in high-risk groups that include older adults and individual underlying chronic medical conditions. But also, as of March 19th, we know that 89 state and public health labs in all 50 states have verified that they're successfully using the COVID-19 test, and actually increasing access to testing is a key priority for CDC.
We've heard some questions about reinfection. Can you talk about whether or not patients who have immunity to SARS-CoV-2 actually are susceptible to reinfection?
What we don't know, this is still a new virus that we don't know on all the immune responses, and we're still learning about how long someone might be immune. We do know for other coronaviruses that people can get reinfected through their lifetimes, and there is some limited information in macaque that we're not able to be reinfected, but we're still immediately reinfected, but we're still learning about the immune responses and how long it may last for this particular virus.
Excellent. Second question, there are those who wonder about mutation of the virus. I know Dr. Whitley and Dr. Gerber, you both talked about influenza, where there's a good deal of change of the virus over time. Can you talk about the stability and evolution of this virus from when it was first isolated in China to what we're seeing now in the US? Are there any differences? And Dr. Whitley, did you hear my question? Okay.
Interested in the development of antivirals for treating SARS-CoV-2. In that direction, then, I remain persistently concerned that we'll see resistant virus appear in the community to one of the two therapeutic approaches that's under evaluation, and certainly the newer therapeutic approaches that will be in evaluation, and it's going to be the responsibility of laboratories who study this virus to determine whether or not mutations have occurred. Obviously, it's too early to know whether that's the case now because selective pressure has not been put on the virus in the absence of antiviral therapies. We'll know more as the antiviral data roll out the next month.
Excellent. Dr. Gerber, did you have any thoughts on the issue of the virus's mutation, if at all, over time?
I think Dr. Whitley said a few very important things that lead to the importance of monitoring genetic sequences of this virus over time. And we know that coronaviruses in general can mutate and recombine. And I think that monitoring sequences over time is going to be extremely important as potential new antiviral might be developed.
I know that you talked about, Dr. Whitley, you talked about the symptoms and how people can develop the infection in the lungs. Can you talk about the role of antibiotics, if at all, in treating secondary bacterial infections and potentially also the prevalence of those infections in people who go to the hospital with COVID-19 infections?
I don't know the actual data for individuals presenting with concomitant bacterial infection. Perhaps Dr. Gerber does. I would say that some of the clinical trials that are being done involve drugs like chloroquine and azithromycin. Azithromycin is not being used to treat bacterial infection, but it's being used as an anti-inflammatory under those circumstances.
Yes.
And if a patient does develop pneumonia, it's recommended that an alternative antibiotic be administered. Maybe Dr. Gerber knows the actual incidence of concomitant bacterial infection at the time of presentation.
Yes, that would be helpful. Dr. Gerber, go ahead, please.
Oh, no, thank you for the question. It is extremely important to determine the presence of bacterial infections at the time of diagnosis and actually the illness. We're still collecting data about this issue, and right now that is a priority for CDC to understand associated with those types of infections, secondary infections. So that is something that we're collecting, information we're collecting.
Okay. Yes, I think that'll be interesting to people as we go forward and look at the various treatments to help people recover fully. Last question. So what do you think, Dr. Whitley, you actually just mentioned several of the key treatments that are being explored and used. What do you think is the mechanism of action that is making those products work in terms of treating the virus or getting rid of the virus?
The current therapies are using azithromycin in combination with either chloroquine or hydroxychloroquine, are using azithromycin as an anti-inflammatory agent. And there's an ever-increasing body of literature suggests that azithromycin as an anti-inflammatory, not as an antibacterial, can accelerate the resolution of lung disease, particularly caused by respiratory viruses. We have to wait and see what the data teaches regarding the controlled studies of drugs like chloroquine and hydroxychloroquine, with or without azithromycin in the treatment of SARS-CoV-2.
Excellent. Well, I want to thank you both for bearing with us and answering our questions, making your presentations on the clinical as well as the epidemiologic issues. We're going to take a very short break and set up the next panel with the government speakers who are going to talk about the needs and gaps. We'll ask everyone to bear with us. You'll see, I think, a slide that just shows the summit, and then we'll get the other speakers set up and ready to go. Thank you all for your patience, and we look forward to the second half of our summit plenary. Thank you. We're back for panel 2. Wanted to start by introducing Dr. Rick Bright from the Biomedical Advanced Research and Development Authority, BARDA. Dr. Bright, I want to turn it over to you to talk about the work that's happening with BARDA thus far and the gaps that you see that would be helpful if our industry could help. Thank you, Dr. Bright. Please go ahead.
Phyllis, thank you. I want to thank you and especially Bio and all of the membership of Bio, the hundreds of partners that the United States government has joined with across the past decades, across every component of the private sector to not only prepare for and respond to numerous outbreaks, but in particularly the level of collaboration and cooperation that has come forth to work together to get us all through this current challenge of the coronavirus. If you can turn to my first slide. On the next slide, please. I think the most important principle that we all need to keep in mind as we work together to respond to the coronavirus outbreak is that speed is the critical component of this response, and actually a critical component of every response. Even when we have the most advanced medical countermeasures, we will fail unless they are present in sufficient quantities with minimal delay and where they're needed. So with these principles in the next slide, our strategy is focusing around partnering for acceleration, risk mitigation, and the manufacturing capacity, and focusing on the supply chain now. We must leverage existing partnerships, also forge new partnerships urgently, and leverage existing platform technologies that have either already FDA approved or authorization or licensure status or in an enough advanced stage development so we can move very quickly through a development pathway. We must also understand that moving quickly does assume much more risk, and we must actively be thinking of every risk mitigation step up front, prioritizing investments and partnerships that will have the greatest public health impact and the probability of success as quickly as possible. We must think about mobilizing our industrial manufacturing base as quickly as possible. Not every company will have a particular candidate, vaccine or drug or diagnostic, but every company has something to offer. And we're asking all companies to work in collaboration and partnership in ways that many of us have never contemplated in the past. Not only working with government and public-private partnerships, but also working with each other to ensure that we have the domestic manufacturing and testing and response capabilities and capacities that we need to respond very quickly. On the next slide, some examples of acceleration are that we are working in parallel streams using very flexible contracting mechanisms. We're proud to say that with our broad agency announcements and our easy BAA broad agency announcement mechanisms that we put in place over the last years, they're paying off in dividends, allowing us in some cases to put contracts in place or modify other transactional agreements in place in a matter of days, a couple of days to a couple of weeks, instead of the traditional lengthy timeline. We're also working hand in hand with our industry partners and our FDA to look at all forms of regulatory development pathways and regulatory response frameworks to be able to evaluate and make available the medical countermeasures as quickly as possible. And we're working with all academic labs and across all government and industry sectors to share access to core services, including clinical specimens that are, believe it or not, still in a rare supply, and animal models that are ramping up in parallel to our medical countermeasure development. On the next slide, when we talk about risk mitigation, we are highlighting the use of vaccine platforms that have extensive experience in the clinic or regulatory review and approval. And many of these technology platforms are allowed or can move much more quickly than some other vaccine platforms. So we're looking at nucleic acid-based platforms, if it's DNA or messenger RNA, vector-based systems, recombinant-based systems, because we can move quickly, because they are also suitable for tech transfer in a number of other facilities to rapidly expand and scale out domestic capacity as well as global capacity in parallel to their development. For the diagnostic platforms, it's clear that we started with the most experienced workhorse testing capabilities in our country, the centralized laboratories. And we have now been able to expand very rapidly into platform technologies in hospital laboratories. And now you see the announcements coming out very quickly about the point of care technologies. It's important to remember that many of these technologies, as they're approved or authorized for use, are still in limited supply. And we must work together to triage where those diagnostics and supplies are used and needed most urgently, so we can make sure we are treating the people who are in most urgent needs of information or treatment as quickly as possible. On the therapeutic side, we are evaluating currently drugs that are currently approved for other indications or in late stage development for other indications. You see already clinical studies ongoing for remdesivir, some antibodies towards IL-6 receptor, and the news on hydroxychloroquine and chloroquine that are now entering clinical evaluation in our country. It's important to recognize that we're also engaged with global partners across other countries and WHO, where we are also receiving and sharing information as quickly as possible. And it doesn't mean that we are not also moving quickly with our industry partners to identify novel drugs and novel therapeutics to the coronavirus itself. But we believe that many of these technologies or therapeutics that have already been approved or extensive regulatory experience might be a frontline response while we wait to develop the novel therapeutics. One important factor and gap is to think about the supply chains. Unfortunately, many of the supply chains and raw materials do not come from within the United States, and therefore it is becoming increasingly difficult to be able to move those supply chains for the raw supplies needed for vaccines and drugs and diagnostics around the world to the facilities where they're needed. We must also plan for success and plan for success today in parallel to initiating phase 1 clinical studies. Therefore, we must begin to acquire at risk all raw materials for production of the medical countermeasure, but also for administration and delivery of medical countermeasures as quickly as possible, so we can ensure that when we have drugs and vaccines available, we have the materials to scale up large volumes quickly, and we have the ancillary supplies required for their administration as well. On the next slide, domestic manufacturing capacity is critical, and this is another area that we ask our industry colleagues and partners to think about very, very carefully. No single manufacturer that I'm aware of has the full capacity needed to make a large scale, full response supply as quickly as possible out of a single facility. It is critically important now to think about scale out and redundancy, not only in facilities in the United States, but also in facilities around the world, so we can ensure that when we have a vaccine or drug available, that there are multiple facilities able to scale up and manufacture and produce and fill and finish and deliver those vaccines and drugs and reagents as quickly as possible. So it's incumbent upon our industry colleagues to work together to identify excess capacity or unused capacity or capabilities now, and begin forging those relationships now, begin conducting those tech transfer technical aspects now, and begin acquiring the raw materials now that will be needed to produce, scale up, scale out, formulate, fill finish, administer, and deliver large volumes of medical countermeasures as quickly as possible. On the next slide, I want to highlight the collaboration across government. We have established, and we had an industry stakeholder call in the on January 31st. We established a medical countermeasures task force that has representatives across HHS and DOD and USDA and VA and CMS and Homeland Security. And so every relevant partner, and some not even on this slide, who are critically important and to collaborate and work with you and work with each other to make sure that we are solving problems in a redundant and collaborative manner. Looking at the countermeasures task force, we announced in January a critical website called www.medicalcountermeasures.gov. Many of you are already familiar with this website as the BARDA Tech Watch website. We have adapted that BARDA Tech Watch website to our Corona Watch website, and we have opened that portal up for all of our interagency colleagues. And we ask you to please use this portal and this website to submit your information to reduce the level of traffic that's going to each of our agencies at this time, because every one of them is reviewing those submissions to the Medical Countermeasures Corona Watch portal. Please use this, and I assure you that your submissions are being reviewed, and every one of the agencies has access to those, so you do not need to send multiple emails to multiple addresses and agencies to have your information reviewed. The next slide just highlights the activity of this portal. I hope you can see this slide clearly, but we have already had over 1,200 submissions to the Medical Countermeasures Corona Watch portal. And they range in submissions across diagnostics, therapeutics, and vaccines and other supporting technologies. We have already had nearly 100 Tech Watch meetings or Corona Watch meetings where we have talked to the companies in more detail about their specific technology or capability, and we're then able to triage those companies to one of the federal agencies who have open solicitations and supporting funding available so we can accelerate moving those technologies through the submission and review process. Please continue to use this portal and so we can ensure that your review is being seen. If you submit it to an email box at this point, then it is unlikely to be reviewed in a very rapid manner because all of our colleagues in government are receiving multiple emails every hour. My final slide is mostly used to remind you of the Corona Watch website, www.medicalcountermeasures.gov, and also to give you the specific web addresses for the broad agency announcement and the easy broad agency announcement in BARDA. I know my colleagues across government will make their solicitation information available to you as well. And also one plea that if you have people who would like to join the fight on behalf of the government, we are hiring rapidly. We could use support in government, not only to review the technologies, but also to work with the partners. So if you're aware of technical experts out there who are available and want to join government on a temporary basis to join the fight, we welcome those resumes at the usajobs.gov website. I want to close, Phyllis and Bio, by showing you a coin that we had created in January in BARDA. Notice the logo, Saving Lives and Protecting Americans. This represents the 50th FDA approval that we have achieved together with public-private partnerships between BARDA and government and our industry colleagues. This shows that we can work together, we can solve problems quickly, and we can get through this challenge together with each other. Thank you all, and this is your challenge from us to you to join the fight and save lives and protect Americans. Phyllis, back to you. Thank you.
Dr. Bright, Thank you so much. Great information. I think those of you who got the background packet will have seen links to many of the broad agency announcements and other things that Dr. Bright presented. Excellent start. Let me pass it over to Mr. Bryce over at the Department of Defense. Mr. Bryce, I think you're coming in on the phone. Please go ahead and feel free to start.
Good morning, everyone. I really appreciate the opportunity to talk a little bit about what DoD is doing. And normally you would associate my organization with CBRN specifically. But as we have the expertise, we have the assets available, we have also taken on coronavirus as part of our mission for the DoD. There are still other places that are working it, but we're focused in on what you see on our first slide. As we look at the things that we are interested in, I want to mention that we have a lot of partnerships already. OSD, Health Affairs, DARPA, HHS, ASPR, HHS, BARDA, the FDA, Homeland Security, DHS, our industry partners, NGOs, CEPI, we really appreciate their support, and then academia, UARTS, we have Nebraska and JAQ. So you can see what we're interested in. I can tell you that we've also looked at masks, we've looked at ventilators, we've looked at wands, we've looked at cameras. So we are interested in the full spectrum of products. Most of them, I believe, are what you see on the screen are where we're trying to head from a focus perspective. The other ones are more commercial, and how do we get a hold of some of them, especially with the supply chain issues that we see. So we want to make sure that we can leverage the private sector. And from that, I would share that we have opened up 3 OTAs. You do not have to, other transaction authorities, you do not have to be a member of any one of them to submit. And any one of the 3 are all being gathered up. And again, with our BARDA colleagues, we are looking at everything that's in them. Currently, we have over 260 that have come in. We will take any more that want to participate in our OTA and give us ideas of where you could help us in these areas. We'd be more than happy. If I ran down diagnostics, of course, we're very interested in the point of care side of this. We have a few efforts already ongoing. We'd also like to get to some screening mechanisms using diagnostics capabilities. So how do we get to screen individuals in or out for coronavirus? So those are things that we're really interested in. As you know, we have a soldier, sailor, airman, and marine deployed around the world every day. So we want to make sure that we're providing all of them a capability to at least look at and see if their fever is due to flu or whatever else may be, or coronavirus. I also want to make sure that anybody out there that has some early technologies, that we're not leaving you out. We're working with DARPA, and we would work with any others that have early technology that may want to talk about the future and have things for the future. But I can tell you our focus will be on things that we can deliver very quickly and early, the same as what you heard from HHS and BARDA. Ours is very similar, getting a capability out and then finding some things that we might do more long-term after that. So if you're a company that wants to submit, keep that in mind, that we will also prioritize things that are longer term, and maybe reaching back out. I would say that with 260 received in about 48 hours, We expect that there will be some more that will come in. It does take time for all of us to look at these, and we will bend them. What I would also say is we'll keep reviewing those, and as we learn more, we'll be going back to them. So don't think that if you haven't heard anything quickly that you're out of the game. That is not necessarily so. We will keep you posted to whatever mechanisms we can to keep you aware. We are also involved with BARDA's Tech Watch. So all the things that Rick Bright just said are the same for us. I would also share that, next slide, that we have an advanced development manufacturing facility in Ology. You can build those partnerships with Ology at any point. You can go direct as a commercial, or you can come to the government and work it through the government. So our Ology is what we call the DoD Advanced Development Manufacturing Facility in Alachua, Florida. So that is another mechanism for all of you to use if you want to. And we also are working with BARDA on their ADMs as well, and we have some efforts in their ADMs as well. So we're working on biomap effort with BARDA to actually ensure that our ADMs are being workloaded and working. So I hope that helps a little. The last slide, or the next slide, shows basically our regulatory affairs. We have in the JPDO an entire regulatory affairs group, and that group can help industry. It can help the government, and we have a lot of capability that as far as our regulatory affairs and quality assurance that we can help companies with, or that we can engage the FDA with. We have also a public law that was passed recently that allows DoD to have some access to the FDA on a more frequent basis, and to help get things that the DoD needs much quicker. So we have several mechanisms that the team here in the JPDO have the ability to work. And if I could just put up the last slide, it'll show you exactly those points of contact. So if you go to our OTAs, you would find that there are points of contact in them, usually our manager, which is an ATI company, but you can also get a hold of any one of the folks that are on this list. And what we try to do is keep it as personal as possible, so that you will actually send an email to myself or my deputy, or the two folks that are actually doing the drug and vaccine development and diagnostics work, and our FDA regulatory lead. So you have access to the top of the DoD response to COVID-19 on one slide. And with that, I am done. If there's any questions, I'd be more than happy to take them.
You heard from Richard this morning, and although I don't know what he had to tell you, I think you're all at this point familiar with STEPI and its mission to, we were built to respond to situations like we are with COVID-19. So I think many of you know that we had been working on a portfolio of 5 pathogens on the WHO priority list, including MERS, and that when COVID started, we also had a small portfolio of platform technologies that we had been working on and had been working on. And then just as a bit of history and background here, as we saw COVID developing toward the end of December, we started watching this carefully, and when we all came back from work to work after the New Year break, we convened our emergency management team, looked at the situation, and decided that it would be appropriate to reach out to our developers of MERS vaccine and our developers of platform technologies and see if once a sequence was posted, they would start vaccine development. Because of the way CEPI works, and I guess I learned this off from many of you in the audience and from working so closely with BARDA over the years, we had written our contracts with these manufacturers in a very flexible way, so that in fact we were able to do some simple contract modifications or task orders to get them working quickly. So at any rate, we were able to get going with an initial portfolio of 4 candidates very quickly. Once it was clear that we needed to diversify our portfolio, again in early January, we also put out a call for proposals. And in that, as Doug Birx has indicated, we were really looking for vaccine candidates that had been in the clinic already. And we received about 49 applications, pales in comparison to what you guys received for all the countermeasures. But we're able to screen them over a weekend, look at 19 proposals, and advance a bunch to our scientific advisory committee, who in well under a week were able to move forward with recommendations. And again, in a very flexible contracting mode, we're able to put in place almost some early letter agreements to get developers working while we agreed to negotiate over the next 6 weeks in good faith a more fulsome contract. And so that is exactly what we have done so far. If you go to the next slide about the paradigm shift, I think Rick already spoke to this, but only a really fundamental paradigm shift in how we do vaccine development is going to get us where we need to go. And so we have been at CEPI, as I think you heard from Richard this morning, really focusing on speed, scale, and access. And as Rick said, this means that a ton of work is going to need to be done in parallel. And as we see now, even with the first candidate from Moderna, phase 1 trials are being done in parallel with early animal work. And we understand now that the animal work will also need to catch up with that. And so we too are looking a lot at issues about manufacturing at risk, and we'll continue to do that. From our perspective, let's go to the next slide about the paradigm shift. I think it means has 4 major implications. First, it means that we need a much more adaptive development strategy than we have had in the past, and a very early concentrated investment. We've made a fair amount of investment in the enabling sciences to support that, whether it's assays or standards. I heard Rick talk about germ collection or animal models. And that is that they're happy to share those resources and the output of all those resources with the global community and any developer who needs them on the enabling science side. We've also been doing a fair amount of work, in particular around immunopathology, with the Brighton collaboration, and have been developing safety definitions, and hopefully these are things that the world would be able to use so that we can look at the most serious adverse events related to potential adverse events related to COVID vaccine, and understand if it's an issue and try to get ahead of it early. Also very much looking at the supply chain. And as it turns out, we had already, for other work, taken a look at, I think, just about all of the manufacturing capacity in the world. And now very much looking at how to pair each of our developers with scale-up manufacturers, and as Rick indicated, to begin working at risk to scale up manufacturing around the world. And I think we are also very focused at being sure each of our partners has a manufacturing partner outside of the United States, and so that's work that's ongoing. So this means it's a pretty expensive undertaking as well. It's going to involve a lot of tech transfer and other things. And I think along the way, we've all recognized that financing this kind of manufacturing capacity is nobody's responsibility in the world. And so we've catalyzed a whole number of discussions with the World Bank and WHO and others to think about how we finance this scale up of manufacturing capacity. And I'm encouraged that this discussion is going really well. Obviously, there's going to need to be a lot of very proactive debottlenecking, as you can see on the slides, and I've mentioned a number of those things already. And then finally, just in terms of the process, we have a pretty high operational bar. We are working actively and in partnership with each of our developers to look at potential errors, anticipate how we can de-risk them and get rid of the bottlenecks before they become bottlenecks. And then obviously, as we move forward, we'll be making a set of go, no-go decisions about our vaccine candidates as we get more early experience. Let me just go to the next slide. This last slide is a slide that shows our current partners. And you can see that they represent the diversified set of platforms, diversified countries, diversified partnership type. I will say that we expect to announce a couple more partnerships later this week or early next week. And I think at this point, too, we are very gratified now that some of the larger MNCs and others have been in touch. You know, GSK has offered adjuvants for use with any of our partners, and a number of other MNC manufacturing partners, I think, are starting to think about and ask how we might all work together to ultimately get, you know, manufacturing at scale around the world for this work. And finally, to say, well, I appreciate that BARDA and DoD have a U.S. focus. You know, the work that he's doing really has a global focus, and that also means that there's going to need to be some global allocation of vaccine in a fair and transparent way. And so, again, we are trying to catalyze and kick off work to have that happen. It's been fantastic to partner with many bio companies, many parts of the U.S. government, including NIAID and DoD and FDA and CDC, and we're looking forward to more and continued collaboration. We can only do this together if we do it, you know, as a whole, and not have us all competing against different parts. And so we would welcome continued input from many of you who I imagine are on the line, particularly those of you who are able to help some of these companies move forward to complete development, to do scale up and manufacturing around the world, and ultimately get some of these products not only through to emergency use, but through to licensure. It's been a fantastic collaboration so far. It's been great to take what we've learned from so many of you over the years and put it all to work, and we're all looking forward to continued collaboration.
Okay, good morning or good afternoon, everybody. So appreciate the opportunity. Thank Phyllis and the bio organizers. Certainly important to keep all of us talking as much as we can in this rather extraordinary time. I'm going to give a high-level overview of some of the activities at the NIH Vaccine Research Center. So we are part of the National Institute of Allergy and Infectious Diseases. And on the first slide there, what I show is that the mission of our center as an NIH research center is to do research, but specifically leading to the development of vaccine and antibody products for infectious diseases. So we have the ability to do basic research all the way through first in human trial. We've done that for a number of diseases, and that certainly gives us the ability to move pretty quickly in certain areas. But if one goes to the next slide, and you can see that I highlight a red box indicating the importance of early industry partnerships. So as an NIH entity, we can bring things through phase one, and the earlier we can find partners, this could be public-private partnership, it can be sometimes NGO partnerships. So we emphasize that early. Certainly for coronavirus, we have emphasized that and have forged a number of very productive partnerships. On the next slide, I want to emphasize that one of the perspectives that we have had from regarding emerging virus diseases is that we should be as prepared as possible, and certainly rapid response is necessary. But what that's done much more effectively if one can be as prepared. So if one looks even at a very simple list of emerging or re-emerging virus diseases, starting with Ebola in 1976, it becomes pretty evident that we will see emerging viruses causing human disease. And especially because SARS and MERS occurred in the last 20 years and they were coronaviruses, we had chosen to put some emphasis there, along with other areas such as paramyxoviruses and flaviviruses, as virus families where we could likely see a new variant emerge. But with regard to coronaviruses, so we see that in 2019, the SARS-CoV-2 virus emerged. And the point that we would emphasize is that the scientific community's work on SARS and MERS allowed us to more rapidly respond to coronavirus. And so we are actually in somewhat better place than if this new viral epidemic were some other virus family of which we knew much less about. So with regard to coronaviruses, we go on to the slide that shows the VRC coronavirus 2 vaccine response. What we are able to emphasize is areas that are our strengths scientifically, where we can make a contribution to the broader field, and that is in the area of structure-based knowledge to allow optimal design of vaccines, and also design of protein antigens to allow isolation of monoclonal antibodies. And what I would emphasize is that structures of HKU1, one of the coronaviruses, and other later SARS and MERS coronaviruses allowed us and others to look at the sequence of the novel coronavirus, and simply based on sequence, have enough information to begin antigen design. We had established some collaborations with related to the mRNA vaccine technologies as one way to move pretty quickly. Certainly there are others, and as has been already mentioned by Rick and Nikki, that scale up in the capacity to address this epidemic is going to require multiple organizations and private sector entities. But with regard to mRNA, it simply makes the point that structures going back now several years for HKU1 taught us how to make and stabilize the spike protein of coronavirus. That allowed us to move quickly when the novel coronavirus came. And the next slide, which shows the sort of the detailed pictures of the mutations, really just makes the point that because many of these viral spike proteins in the coronavirus family and in other virus families are metastable because they are fusion machines, one can stabilize them with various mutations. And here, for coronaviruses, these stabilizing mutations allow them to be expressed at higher levels. They allow them to maintain structural integrity, and we find that they actually have better immunogenicity. So we have employed those here. The next slide shows that the field investigators, in this case, investigators at the University of Texas, and now others, were able to rapidly solve the structure of the novel coronavirus. And that was, again, engendered by the fact that protein in a stable form could be expressed quickly. So the next slide I would emphasize is the timeline slide. That's highlighting our partnership, in this case with Moderna, on the mRNA vaccine platform. And the slide, I won't go into detail, but at the very bottom there shows the 65-day timeline from sequence to first human injection in a phase 1 study. And we mentioned this just to emphasize that this is something we've been working towards, others have been working towards, which is how can one quickly move from vaccine design to phase 1. And there are a lot of pieces to that. There are regulatory pieces where the platform needs to be seen and understood by regulators, and the regulatory aspects and safety aspects of that platform need to be understood. But in the right circumstance, one can move quickly to human trial. And then the next slide, I just want to make the point of the multilateral support that's really engendered the mRNA vaccine platform in general. And that goes back to early DARPA funding into the research and development. In the particular case of the work that we're doing, CEPI has played a role in funding. The NIH has played a role in funding the phase 1 study. BARDA has a role to potentially play in advanced development funding. So I think, as others have mentioned, this is going to be a multilateral effort between the public and private sectors and NGOs in order to move this and many other vaccines forward. I just want to end with a word on monoclonal antibodies. So that's a slide, if all can see it, it's just entitled Monoclonal Antibodies. I think there's an emerging consensus that there's the potential for both therapeutic and prophylactic, immunoprophylactic indications for monoclonal antibodies. And some of the same principles apply here, that if one understands the structures of the proteins well enough, one can rapidly use them to isolate antibodies. DARPA and other organizations have funded rapid antibody isolation platforms. There are new technologies which really engender rapid monoclonal antibody isolation from antigen-specific B cells. We have also forged some partnerships in that arena, and are working with several groups to help them rapidly isolate antibodies so that they can then move those forward in development. So I'll stop there and thank all of you for participating.
My name is Julie Kearney. I'm the Chief of Staff at FDA's Center for Biologics Evaluation and Research. I'm sorry that Dr. Marx was made for join for this call. As you can imagine, it's all hands on deck right now at FDA with the COVID-19 response, and he had to attend to some other matters. So FDA's mission is to protect and promote the public health, and we do that in part by ensuring the safety and effectiveness of the medical products we regulate, and by helping to expedite their development, particularly in response to public health threats. At FDA's Center for Biologics Evaluation and Research, we're involved in several aspects of the COVID-19 response, including ensuring the continued safety and availability of the blood and plasma supplies, the development and approval of certain therapeutic products, such as convalescent plasma and hyperimmune globulin, and the development and approval of preventative vaccines against this novel virus. At the same time, the center also has to ensure the continued availability of key medical products manufactured from plasma, such as immune globulin and other plasma derivatives, for which the U.S. is a leading global producer, as well as ensuring the timely delivery of important vaccines, such as the approximately 160 million doses of next year's seasonal influenza vaccine. Our work certainly includes working closely with our colleagues across the FDA and collaborating as much as possible with our federal partners at BARDA, DoD, NIH, and CDC, international partners, NGOs, and other stakeholders. Just wanted to highlight a few things that we're doing associated with the COVID-19 response. And although it's not directly in Bio's domain, it might be useful for you all to know that we're now facilitating access to convalescent plasma for the treatment of COVID-19 disease through an emergency IND mechanism based on data that's emerged from China. Although participation in clinical trials is one way for patients to obtain access to convalescent plasma, these might not be readily available to all patients with potential needs. So this is especially important in places like New York, where there's simply not resources available to conduct a full-scale clinical trial. With that in mind, given the public health emergency that the expanded COVID-19 outbreak presents, while clinical trials are being conducted, we're facilitating emergency IND access. And in fact, we just this morning posted a website to provide information to providers on how to submit an emergency IND for convalescent plasma for treatment of single patients, so that while clinical trials are being conducted and data is being generated, we have these access mechanisms in place. So we know that we need to balance patient access here with the continued collection of data to support potential licensure of products, which will ultimately benefit the greatest number of patients. So we anticipate that a protocol that we're developing in conjunction with our government partners will soon simplify the availability of safe, high-titer plasma units for this purpose through an intermediate-sized investigational new drug application protocol held by a U.S. government public health agency. Moving from convalescent plasma to the manufactured therapeutics front, based on preliminary data indicating potential efficacy of antibody-based therapies against this coronavirus, we're actively working with several manufacturers to accelerate the development of hyperimmune globulin preparations for study. We're working to facilitate necessary manufacturing work, and in fact, anticipate that some sponsors may have product available for clinical trials within 4 to 8 weeks, which is a remarkably accelerated time frame. We will be using maximum regulatory flexibility at FDA to try to get these into the clinic for study before July. With respect to vaccines, we're working with manufacturers to maximally accelerate vaccine development. I'm sure we'll hear more tomorrow about the range of different vaccine approaches, ranging from those that are mRNA-based to live virus vaccines. That said, we do need to proceed with some caution here because prior coronavirus vaccines have been associated with immune enhancement, which is when vaccinated individuals subsequently infected with the virus develop worse disease than if they had not received the vaccine at all. Although the need here is urgent, we still need to exercise care that what is developed is both safe and effective and in keeping with FDA standards. In conclusion, like many others, we look forward to working through this crisis with the help of our partners such as you in order to come out on the other side as healthy and as soon as possible. Thanks so much.
Thanks everyone for your great comments, for all of our presenters with your excellent information for the companies. We want to actually move forward with the collaboration that I think has been encouraged by all the speakers all day today, from Ambassador Birx all the way through to Julie Turney at the FDA. So what will happen now is that we will encourage everyone to start thinking about the treatment, prevention, and diagnostics working groups. Those groups will be meeting through a webinar series this afternoon, tomorrow, and the next day. Many of you have joined. Some of our government colleagues will be joining us as well. And I think the goal is to have detailed conversation about what we can be doing together to speed the development of diagnostics, treatments, all the different kinds of treatments and vaccines, so that we can bring products to serve the public health need for this very important global health emergency. Thanks everyone for your patience, for all of our speakers for their great comments and slides, and we look forward to working with all of you over the coming days and well into the future to solve this emergency.